Now I will discuss the topic of splicing of pre-mRNA. Splicing is basically editing or cutting of pre-mRNA. It is process of cutting or trimming the pre-mRNA. Then pre-mRNA is transformed into mature mRNA in this process after splicing. When snRNP is altered or there are some changes happen to snRNP protein, it can lead to spinal muscular atrophy. And spinal muscular atrophy can cause weakness, hypotonia, developmental delay, and loss of respiratory muscles. Due to hypotonia, it can cause floppy baby syndrome. Some antibodies are against snRNP protein. It is highly specific for SLE or Systemic Lupus Erythematosus. We know that SLE presents with fever, rash, joint pain. So antibodies that are against snRNP, they lead to SLE. Splicing of pre-mRNA. It is part of process by which precursor mRNA, that is immature mRNA or pre-mRNA, is transformed into mature mRNA. Introns typically begin with GU and end with AG. So this is GU and end with AG. This is introns. Introns are basically removed in process of splicing of pre-mRNA. Splicing its meaning is editing or cutting of introns from precursor mRNA. Alterations in snRNP assembly can cause clinical diseases. When snRNP assembly or alterations happen and into snRNP assembly, then it can cause spinal muscular atrophy. snRNP assembly is affected due to decreased SMN protein. When SMN protein is decreased, then snRNP assembly is affected. It can lead to congenital degradation of anterior horns of spinal cord. When anterior horn of spinal cord, congenital degradation of anterior horn of spinal cord, that lead to symmetric weakness, hypotonia, or floppy baby syndrome, that is congenital degeneration. snRNPs are snRNA bound to proteins. For example, Smith protein. These proteins form Spliceosome, that is cleaves pre-mRNA. Anti-U1 snRNP antibodies are associated with SLE or other mixed connective tissue diseases or other rheumatic diseases. So this is U1 snRNP. This is exon 1 and this is exon 2 and this is intron site. So here is spliceosome happen and then mature mRNA is formed. Mature mRNA contain exon 1, exon 2. It cleaves at 3' splice site